So, chapter 3, we talk about biological macromolecules. So, expanding on chemistry, where do we go from there? Well, we talked about 4 different kinds of macromolecules. Things I want you to be familiar with all of them. I mean, some have more of some of these categories than others, but all of them are constructed of monomers joined together to be polymers. Have an idea what the monomers are called and the polymers are called for all of the 4 macromolecules. It's saccharides or carbohydrates, lipids, proteins, and nucleic acids. So know what their monomers and polymers are each called. Does the bond that connects them have a special name? Most of them do. Have an idea. Most of the time, we're most interested in the polymers. What are kind of some examples of some polymers? What are some uses many of the polymers have? A rough idea of their structure. I mentioned down here, for example, there's some differences between DNA and RNA. RNA has uracil, right? So what does DNA have that in place of uracil? DNA has a different structure than RNA. Right? So DNA forms double helixes, where RNA normally does not form a helix, but it's because it has a different purpose. Okay, so just for example, again, not going to spend too long here. Just if anything, if you're not familiar with one of these categories for carbohydrates, lipids, nucleic acids, or proteins, then make a note of that. Okay. All right. We use microscopes. Chapter 4, we talk about cells, and I just mentioned briefly there'll be some microscope questions on there because we use microscopes to observe cells. There are different kinds of microscopes. We have light microscopes. We used compound light microscopes in lab. Right? So it's a compound because you've got a lens in what we call the ocular lens. That most of those were 10 times magnification, and then you had different lenses. Different lenses down on the rotation. You had a scanning, then a low power and a high power that we could use. So it's compound because you've got a lens in the ocular that magnifies, and then that increases the magnification of the lower lens down below. Those are compound light microscopes. There are different kinds of electron microscopes that, again, we can't put living things in electron microscopes because those are in a vacuum or they've got to be at very low temperatures. But anyway, just some basics there. Cells. So there are prokaryotes and there are eukaryotes. What is the difference? There are some structures and or organelles that are found in both prokaryotic and eukaryotic cells. What are those? And then I gave you a list of common organelles found in animal cells. I've asked you to identify organelles found in most animal cells. I've asked you about there's like 3 organelles typically that you would find in plant cells that are not in animal cells. Know what they are, know what their uses are. If I gave you a picture, could you identify them? And again, just an idea what each one does for and within the cell. And then the plasma membrane is one of those organelles, and that's one of the ones that shows up in both eukaryotic and prokaryotic cells. And the fluid mosaic model is the prevailing theory we use to describe this mosaic that composes the membrane. It's mostly these phospholipids joined together, but there are also various proteins. There are cholesterols in there to strengthen and stabilize the bonds between the phospholipids. There are proteins that there's some proteins with carbohydrates on the outside, cells use to identify each other. There are proteins within the cell wall or the membrane that we use for transport of materials in and out of the cells. So let's be familiar with all of those. Okay, and that's not a bad place to stop. So we're going to stop there.